Hello everybody. So in this video I'm going to go over basically a pre-lab lecture for your lab, your drug chemistry lab that you're doing this afternoon for this virtual lab that we're doing since I'm not there and I'm still in New Orleans. So what you see on the screen right now is actually the student view of our lab, our class, and as you scroll down you will be able to access the lab under module 10, I think. So let's go down to module 10. And so there's module 9, and then there's crime scene, exam 2, and there we go. There's module 10. So we already did this drug lecture, this drug analysis lecture before I left, and so there'll be a quiz on it. Don't forget to do that quiz after you do your toxicology lecture from earlier today. But here's all the stuff that you'll need for your um, the drug chemistry lab that you'll be doing for this lab virtually. It's not in here yet, but it will show a video there. You can get to the video using the module as well. So just make sure you're checking that because the video there will be there for you to watch as well inside this module. And so the first thing to do is you'll download the drug chemistry lab virtual. That's the handout, and then the drug chemistry test results. And you'll use these together to do the lab. And for this lab, since it's virtual, we'll do it. We'll have the grades be individual. This will not be a group project. You can do it individually. And so if we scroll down a little further, we have our toxicology. That's our lecture that you should do this morning, right, before you even do this lab, and the assignment will not be till the weekend, as always. So click on and download both the virtual lab and which is the handout, and the drug chemistry test results. And what you should get are two PDFs that look like this. Open up like that right there. So the first one you'll see is this right here, which is the handout. And then the next one you'll see is this right here, which is all the test results. All right. So normally you would be doing this in lab, doing this experiment yourself in lab. But since it's virtual, you'll just have to look at pictures of the test being done. And these are the same kind of colorimetric tests that you would do in real life out in the field when you're trying to test for different drugs and stuff like that. Okay, so let's go through this first. If you look at this is just background information. I recommend reading it. It's not absolutely necessary, but some questions might be answered by reading this material right here. I'm not going to make you read it though. So it talks about over-the-counter drugs like aspirin. You are testing things like aspirin, and then Tylenol, which is acetaminophen, and then citric acid. So we're all doing that kind of stuff. Then we get down to the controlled drugs right here. And so hopefully when it prints out for you guys, it looks a little bit better than that. But I mean, you don't really need these molecules. Just kind of show you stuff. We'll scroll down some more. This is tetrahydrocannabinol. That's the active ingredient, or THC, in marijuana. So read about that too. And it talks about here how if you hear that one, the test from marijuana in the field, it's called the Duquin-Levine test. And I talked about that a little bit in the Watchman B lecture, the drug chemistry lecture, right? Heavy metal poisoning, stuff like that. This talks about RF factor, or RF factor. This is also known as the retention rate, or the retention factor, or the flow rate, or the rate of flow, or the retardation factor. That's when you're doing that chromatography stuff I talked about in both the paper chromatography, or the TLC chromatography, that I've talked in both the drug chemistry and the toxicology lab. But you won't be using it in this lab, so don't worry about it. So some vocabulary to know about, and then references. That's great. So this is the very first lab right here. And so what you're going to be doing is you're going to be going step by step through the procedure and reading the procedure. And as you read it, you'll see that it's already been done for you, basically. So, for example, it says right here, use a pencil and label this row A for aspirin. And so if you look over here, we already have this here. I don't know why I got cut off right there, but this says experiment 1, and then I'll fix that. But see here, here's the row, the first row right here, and the first row is labeled aspirin, right? So just like it says in in step 2, right? And then step 3, it says label this row for S for Alka-Seltzer. Then you go back over here, and it's Alka-Seltzer, right? Next one, sodium bicarbonate, B for sodium bicarbonate. We just labeled it the whole name for you, sodium bicarbonate, and so on, right? And so what's going to happen then is when you go back to the lab handout, this is experiment 1 for over-the-counter drugs. So you'll be testing a whole bunch of over-the-counter drugs, as you can see. And then, just follow through and read the steps, but they've been done for you already, right? And then the thing that's kind of weird is that you have to scroll all the way down to the bottom, and that's where your data sheet is. We made a data sheet for you right here. And so here's the data sheet right here for experiment 1. And so if you look at it, it asks for the initial appearance. So you look at the picture, aspirin right there is a white powder, right? So I would put white powder there. And then it says what's the reaction with water. It doesn't have any reaction with water. So when it says reaction with water, usually it means does it make any bubbles or does it turn different colors, right? And so for aspirin, it doesn't do anything, so I'll put no reaction there in this one. Then it says universal indicator and pH. That's based on a color, so we added universal indicator to the aspirin, and it turned this bright red orange color. How do you know what pH that is? You scroll down to the bottom right there, and here's the chart right here. So that bright red orange, that's red orange. So I would go less than 3 pH, right? So that's actually very acidic. Anything below 7 is very acidic. So universal indicator for aspirin, go back over here and just label as the color being red or orange red, and then the pH being less than 3, as we saw on the chart. Next, it says reaction with hydrochloric acid. This is another thing that's just almost like water. The reaction here is whether or not there's any bubbles formed, and if there's bubbles, that's a reaction, right? They also call that effervescence, if you were wondering, making bubbles. But aspirin doesn't seem like it has any bubbles, so don't worry about it. It's kind of hard to tell. Like this one right here, maybe sodium bicarbonate right there has bubbles, but if you need to, you can always zoom in. Just do your best, but you can always zoom in right here. You can actually see that Alka-Seltzer makes bubbles. See, with the water, you can see that's how bubbles are. The powders you can't really see through, right? But like bubbles, you can see through. So if you look at hydrochloric acid right here, it doesn't really do anything with. It doesn't make any bubbles with the aspirin. The aspirin just sits there. But if you go down to sodium bicarbonate, you can see clear bubbles there. So that's what when they ask for a reaction with hydrochloric acid, they're asking you if there's any effervescence or bubbles, right? So you indicate that for HCl and aspirin, there really wasn't anything at all. So we would say that would be no reaction. And then I'm zooming out so we can see the whole thing again. And then over here, we did that one right there. So for aspirin, it would be no reaction. Then it says reaction with ferric nitrate, and you can see here it actually did have a reaction. It turned purple. So in this column right here for ferric nitrate, to identify whether or not there's a reaction, it's either going to be a color change or some bubbles, right? So you can see that color change and bubbles, and so that will tell you whether or not there's a reaction. So over here for ferric nitrate for aspirin, you would write, is there a reaction? And you go yes, because there was a color change. And you can even indicate that it turned purple if you want to. And so basically you go through experiment 1 and just look at all these different colors and all these different wells right here and fill in all the blanks on the worksheet, because they all correspond to the blanks on the worksheet, right? And it says right there NR for no reaction, right? So then it says what is the identity of unknown A, what's the identity of unknown B, what's the identity of the unknown crime scene. So what's happening here is that when you look at these test results for unknown A, B, and crime scene, they should match something from up here, right? And if they match something from up here, then that means that's what that was. So that's what the unknown is. For example, I'll do unknown crime scene unknown for you. See, so it's a white powder, and so everything has a white powder, so that's not very helpful. And then does it react with water? It doesn't react with water. Most of them don't react with water, except for Alka-Seltzer. And then when you add in when you do universal indicator, it's actually a green color for this one, right? And so green was down here, like between 7 and 8. And so let's see if there's any of these known ones up here that are green. So sodium bicarbonate is actually green, all right? So that's good, and it's the only one that's green. So it's probably sodium bicarbonate, which is the unknown. And then the the one with hydrochloric acid, it's hard, it should show it shows bubbles for sodium bicarbonate, right? So let's go back to unknown crime scene, and it also show bubbles, right? So you can notice here that, and then the last thing, an iron ferric nitrate. This unknown turns brown, and so this sodium bicarbonate also turns brown. So the unknown crime scene here is obviously sodium bicarbonate. So now you guys do the rest of them. Like you can do unknown A, you can do unknown B. All you're trying to do is match the entire row of test results with something that's in the knowns to figure out what your unknown is. Okay, and so that's how you fill out the bottom part right here. So unknown A, unknown B, and unknown C. Crime scene unknown. Then the next one is going to be the control drugs. So here we have LSD and marijuana. It asks the question under observation, under UV light, does it react? Like what do you see? Does it glow? So this is like whenever you go bowling or something, or to a club, and your shirt glows, right? Things that are UV, UV active, they'll fluoresce under UV light, and so they'll glow, just like a white shirt will under a UV light or a black light at a club. And then the other one is the reaction with HCl. And again, if it's a reaction with HCl, it's like, is there any bubbles, right? And so we'll do that one with these two right here. We scroll down a little bit. So here's LSD right there, and so it's initially a white powder. So you can put there's not a space for it, but it's a white powder. So it's asking you under UV light, what does it look like? This definitely kind of glows under under the UV light. It would be completely black if it didn't glow. So it definitely glows under UV light. So observation under UV light, it glows. And then reaction with HCl, this one right here, and it kind of looks like bubbles, but it's not. So it's not bubbles. And so it doesn't react. And then you can see marijuana here, and that's marijuana, and then that's marijuana under the UV light. Acts the same way as LSD. So both of these under UV light, they both glow under UV light. But you can go in and do it yourself and see what's going on there. And then here's marijuana with HCl. I'll let you figure out whether or not you see bubbles or not in that one. So do any of these things for powders or drugs fluoresce? Answer that question based on these test results. And then what's the name of the marijuana test that we do in the field? And so that was up in the reading, so you have to go look for that. For this one, the next one is going to be presumptive test for cocaine. I mean, basically we're doing the exact same thing again. You scroll down here, right, and here's all the different, I don't know why these numbers are getting cut off. I need to fix that. But this is experiment 3 right here. It'll be fixed on your file. So here's the befores of basically all these white powders. And then the cocaine is usually tested with Scott's reagent, right? And so cocaine is positive when it's positive for cocaine whenever it turns blue with the Scott's reagent. So you see there's some things on here that do not turn blue, right? So those are probably not anything that's cocaine-like. So just go through and answer all those the same way right here too. So name of cocaine sample ID number, just write in there unknown, crime scene unknown, quinine. Let me see something real quick. Okay. Crime scene unknown. Where is it at? I lost it. A lot of stuff got cut off here. I need to fix that. That's okay. I'm just showing the pictures anyway. So crime scene unknown, and then it's is going to be the ID number, right, for the first one, and so on, right? Quinine, unknown A, unknown B, unknown X, right? All those fit, right there. Description of the original sample right here. So all of them are going to be white powders. And then here, what was the reaction with Scott reagent? If it's pink, then it's no reaction, but if it's blue, then it has a reaction with Scott's reagent. And so it's asking you which of these samples could be cocaine, because cocaine turns blue. We're going to actually skip the fourth one, We're not going to work on that one at all. So, yeah, we're not going to work on the fourth one at all. So you can skip this one, X that one out. You don't have to do number 4, experiment number 4. And then the last one is experiment 5. And you're doing the same thing you did in experiment 1 as in experiment 5. And now you're really going to identify what the drug is. What is unknown X and unknown Y and unknown Z. And so you go here, and then we'll scroll down, and you'll see unknown X, Y, Z. And then also LSD is down there too, because LSD is down here too. So appearance under UV light, does it glow? And so this is what I'm talking about. If you can see it glowing, you'll see the drug. Whereas if it doesn't glow, it's like completely black. Like look at unknown Z, it's completely black. So go through and answer those questions. Again, any reaction with water, usually is there any bubbles? What is the universal indicator color here? So we have like red again. We have this kind of yellow-green, so that's actually, oh sorry, yellow-red, or mostly yellow, so that's actually a little bit up here, right? Another one like that too. And then reaction with HCl, again, is are there any bubbles? Reaction with ferric nitrate, is there any bubbles, or is there any color change? And then also Scott's reagent, right? And then Scott's reagent will remember turn blue when it's cocaine. Okay, so it's a pretty easy lab. Just go through and answer those questions. Okay, and fill in all your stuff from these colored results into your chart right here, and then you should be able to identify using those colors, using those test results, which one is unknown X, which one is unknown Y, and which one is unknown Z. So for your lab report, your items are going to be the unknowns, right? So there's only 4 unknowns, I think. I believe it's unknown X, Y, and Z, and then there's the unknown A, B, and then crime scene unknown. So there's actually, I guess that would be 6. Yeah, that's 6. Yeah. So yeah, that would be 6 unknowns. So in your lab report, just write in there item 1 labeled, unknown A, right? That's one of your, and then item 2 is unknown B, and then item 3 is unknown, is crime scene unknown. Item 4 is unknown X. Item 5 unknown Y, and unknown Z is item 6 is unknown Z, right? So you should have 6 items, and then your results section will show what you determined each one of these unknowns to be. And so again, just to remind you that this is a single person lab, individual lab. You do not have to work as a group. It's virtual. And then so that's it. Good luck, and let me know if you have any questions. We'll go over what the right answers are when I come back on Thursday. Okay, thanks. Bye.